Hi, this is Tiffany Martin with Medical Microbiology, and we are in Chapter 5, and we just covered the electron transport chain. So now we need to do a little bit of review from the electron transport chain and then some summary of cellular respiration. So we finished up with the diagram. Hopefully you've gone through that several times if needed to understand what is going on. These are some of those concepts I keep talking about talking out. Got to talk these out to people, okay? If you can't get in a study group, talk it out to your roommate, to your dog, to your mom, whoever you can sit and listen to, talk it out. Okay, so the electron transport chain process of chemiosmosis is occurring at the electron transport chain. We know it's a cell membrane. We know that there's oxidation of the food is bringing these electrons. So we saw how those electrons come in and how they get out. So see how far you can get on this. Okay, so the electron transport chain is a series of electron carrier proteins in the membrane. The electrons are passed and they lose energy. So they're losing energy, just like our potatoes from the fire, they lose energy. At the beginning, they're considered high energy electrons, but after being passed, their energy is used up. This energy is used, they transfer these electrons to the next carrier, and the energy is what is used to pump those hydrogens across the membrane. So those proton pumps use that energy from these electrons to push or pump those hydrogens across the membrane. Ignore that, go back to page 4. That's what we just did with that video of talking about where these electrons are going. All right, so we just did that. You should have a great drawing of all those things we did. Again, it helps if you draw it out. So this is a book's figure. Actually, I don't know. It may be the book picture from your book, but there's your membrane. And in bacteria, this is your prokaryotic membrane. There is your proton pump. You've got NADHs coming in here. Those hydrogens, those electrons are stripped from that and are accepted by these integral proteins. From there, they take the electrons, and that generates enough energy to pump these hydrogen ions across. Remember, these NADHs come from glycolysis, Krebs cycle, and then all those other pathways that we didn't really talk about when I said that there were Plan B and Plan C. The book covers those. Again, if you've got biochemistry in your future, it's worth your while to take a look at it, but I know it's summer, so I know you guys aren't going to do that. Anyway, NADH becomes NAD+, and he goes back to glycolysis and picks up more electrons. Hydrogen ions come out. They are desperate to come back in so that ATP synthase can phosphorylate ADP to make ATP. We've got to keep these electrons moving so we can keep this process going. And so these electrons get passed from one protein to another until they have lost all their energy, and we need to get them out of the electron transport chain. And here comes oxygen, 1/2 O2 singlet oxygen, which is very reactive and wants to steal electrons. And this is great because we want to get rid of electrons. 1/2 O2 picks up 2 electrons. There's extra hydrogens that have just come through this proton channel. They also will join with that, and we make the byproduct of cellular respiration. We make water from this. Okay, one thing I didn't tell you, do you remember that guy FAD? FAD picked up some electrons in the Krebs cycle and became FADH2. He also is bringing his electrons to the electron transport chain, but he can't drop them off at the very beginning. The way he's shaped, it just doesn't fit. He ends up dropping his electrons off a little ways in, and you can kind of see that compared to NADH. So he drops his electrons off a little bit further, and then his electrons go through as well. So they're useful, they get energy from it, but because he didn't go through the entire line, we don't get as much energy from it. And this is going to be important because for every NADH, we can produce about 3 ATP molecules. Now, it's not perfect, but it's about. For every NADH, we can produce 3 ATPs. For every FADH2, we only produce 2 ATPs. So that's going to be important because we need to kind of keep up with what's going on. All right, let's see if you can fill in the blanks. Go for it. How'd you do? So what's the final electron acceptor? Oxygen. That's why this whole process is called aerobic respiration. That's where it comes in. Now, remember I said if you can't make ATP, you die? There are some chemicals that can block this final electron transfer to oxygen. Cyanide, have you heard about cyanide? Cyanide, if you've heard about it before, it's probably in a like an old James Bond movie or something, maybe a new one. Where the secret agents have a capsule of cyanide and they hide it. And if they were ever captured, they're going to take that cyanide. It's a fast-acting poison and it'll kill them so that they don't have to give up their secrets. That may be how you've heard of cyanide, but cyanide is a chemical, and what it does is it blocks the enzyme that's responsible for that last transfer of electrons to that final electron acceptor, oxygen. So it stops that completely in every cell in your body. If you don't make ATP, you die. So cyanide will block that process, and that's why it's a poison, that's why it's so toxic, and that's why it's so bad. I may post well, I'll see about that. Anyway, let's go back to this. Cyanide will stop that. Let's see what else is going on. Oxygen accepts the energy in pairs because that's how many electrons it can take with its valence shells. Hydrogens also are involved, and 2 hydrogens plus the 2 electrons plus the 1/2 O2 gives us water, and water is a byproduct. During chemiosmosis, every NADH gives us 3 ATPs, FADH2 gives us 2 ATPs. Now, we call this whole process oxidative phosphorylation. If you remember back to glycolysis, I mentioned that we needed to add in ATP, and those phosphate groups were added to the glucose. That is substrate level phosphorylation. You're phosphorylating, you're adding a phosphates to a molecule. The substrate phosphorylation is adding it to glucose. This is considered oxidative phosphorylation because all of those oxidation reduction reactions, all of the energy needed to phosphorylate ADP comes from the oxidation of food. And if you remember, we completely oxidized glucose. We got rid of all of the molecules in glucose. Very efficient process. Okay, so it's checkbook time. We need to account for everything. The ATP yield from complete oxidation of glucose in a prokaryotic cell. So if you go back to each of these steps, how many ATP did you make? But you also need to keep up with NADHs because we know for every NADH, we get 3 ATP. For every FADH2, we get 2. So glycolysis, 2 NADHs, we actually made 4 ATP, but we do need to put that subtraction, that debit column. We use 2 from there. All right, so prep steps, no ATP was made, but we did have some oxidation reduction. So we have some oxidation occurring. We had 2 of those. Krebs cycle, if you go back and count, during the Krebs cycle, there were 3 times in the Krebs cycle where we made NADH and 1 time where you made FADH2. Go back and look at that picture. You really need to. I should have showed you at that time. There was 4 times that oxidation reduction occurred. 3 times they made NADH. 1 time we made FADH2. But you remember we have to put a big 2 in front of everything. So per molecule of glucose, which is what they're asking for here, per molecule of glucose, we made 6 NADHs and 2 FADH2. Now we're going to do the math in this column. For every 1 NADH, we make 3 ATP. So 6 plus 2 plus 2. I have 10 of these. Times 3 is 30 ATP. I have 2 FADH2, but I only get 2 ATP from those. So 2 times 2 is 4. So I can add that in this column. And if I add this all up, I make 40 ATP by chemiosmosis. I do need to subtract the 2 that I used at the beginning. And so for prokaryotic aerobic cellular respiration, we net about 38 ATP. Okay, if you remember at the beginning, I said there was 36 or 38. 38 is what prokaryotic cells do. Eukaryotic cells only net 36 ATP. What's the difference? A eukaryotic cell, remember glycolysis occurs in the cytoplasm. Where does the prep steps and Krebs cycle occur? The matrix of the mitochondria. We need to get the pyruvic acids, the end products from glycolysis, into the mitochondria in a eukaryotic cell in order to undergo the Krebs cycle and the electron transport chain. It's going to take some energy to get that pyruvic acid into the mitochondria. Guess how much energy you use there? 2 ATP. So that's where that difference comes from. You're going to expend a little bit of energy to get that into the mitochondria. Prokaryotic cells don't have to do that. So that's what's going on there. Okay, that was a big, huge chunk. Go through it, ask me questions. I will be online, WebEx on Thursday, and open for questions. Talk it out in your study groups, and this is going to take a lot of talking out. This is a big thing, and get that going. Okay, I hope that helps, and I'll see you at the next video. We're not done with the chapter. Still got to do fermentation, so join me in the next video.